Good afternoon to all the virtual audience, and thank you for joining us this week's seminar from the Living Earth Collaborative and Washington University's Evolution, Ecology, and Population Biology Group. Today, I have the pleasure to present Dr. Alex Linan. Currently, he's a postdoctoral fellow at the Center for Conservation and Sustainable Development in the Missouri Botanical Garden, working under the supervision of Dr. Sebastian Tello and Dr. Christine Edwards. Dr. Linan earned his bachelor's degree in biology and chemistry from the Missouri State University, and his PhD in biology as well from St. Louis University. Likewise, he holds the prestigious National Geographic Explorer title. Dr. Linan is building an impressive profile of publications in prestigious journals, such as the Molecular Ecology and the Annals of Missouri Botanical Garden. And there are two interesting facts that from Dr. Linan that I would like to share with you. So first, as an undergrad, he started doing science with mussels, but they were too mobile for him. So he was looking at other organisms, and then he was looking for some organism that he was less mobile and decided to work with plants. So that's the history, how he got involved with plants. And the second fact is that he spends his free time removing honeysuckle, this invasive plant, from all these different parks of St. Louis. Dr. Linan's research interest is focused on conservation genetics and molecular systematics, as well as the understanding of the hybridization processes in plant communities. For his current research, he's working in two of the hottest biodiversity hotspots in Earth. I'm talking about Madagascar and in the Andes. Before I hand it over to Dr. Linan, I want to remind all the audience to please leave the questions and comments in the YouTube chat section. Without further delay, let me introduce Dr. Alexander Linan and his presentation on his recent work entitled From Islands, Trees to Mountain Forests, Exploring Parents and Drivers of Diversity Within Biodiversity Hotspots. Take it away, Dr. Linan.
Thank you very much, William, for that introduction. And thank you to the LEC for inviting me to give this talk. So as the title suggests, I'm broadly interested in studying diversity in some of the most biodiverse areas on Earth. So this is a map of the 20 or so biodiversity hotspots. And these are areas that contain a high amount of endemic flora and fauna found nowhere else on Earth. My work seeks to characterize this diversity and understand how it arises. And today I want to show you my work in two of these hotspots, the Andes and the Madagascar region, including its surrounding islands, and looking at tree diversity at two different levels. So first, I want to talk about species boundaries of a group of very closely related tree species in the islands surrounding Madagascar. And then I'm going to shift gears to look at a much more bigger picture diversity at the level of entire forest communities in the tropical Andes. So first, let's dive into the Madagascar region. So I guess before I go too deep into that, oftentimes many of these biodiversity hotspots have a lot of sympatric, closely related species. And oftentimes it can be very difficult to delineate species boundaries between these, particularly in tree species that tend to readily exchange genes and experience lots of hybridization and integration. So typically, taxonomists will describe species on the basis of morphological characters that are differentiating groups of individuals. However, in trees, this is much more difficult because, as I said, they experience a lot of gene flow and can create a lot of hybrid individuals that display intermediate morphologies. And a really good example of this is in, probably the best example is in oaks. So that's pictured here, in which case we have one oak species. I think this is Quercus rubris on the right, and another one on the left, and then their hybrids in the middle, displaying a very nice intermediate morphology, which if you're a taxonomist out in the field, and hybrid zones can be very big geographic areas, it could easily be very confusing and difficult to determine when one species starts and another one ends. So these patterns of hybridization are kind of broadly reflective of a speciation continuum model, whereby populations maybe start out as panmictic, where there's little to no reproductive isolation. And under this scenario, of course, we'd expect very little genomic differentiation. However, given time and differential selection forces, we can expect, we can maybe see a situation where there is limited genomic differentiation, but these two groups are still experiencing relatively widespread gene flow. And this is a case we see in many plants or many tree species, in which case we have a kind of balancing act between the homogenizing forces of gene flow versus the differentiating effects of divergent selection. So in many tree species, we may have morphologically different species, but perhaps they're not very genomically differentiated, in which case we might just see a few genes that are very strongly selected as being divergent, driving the morphological divergence that we see that explain the morphology we see. So, and of course, under maybe ideal situations, ones that you rarely see in trees, closely related, we have complete reproductive isolation with very strongly diverged genomes. And this is maybe a case we don't see very often in trees. So an example or an instance in which we can have, we can be looking at patterns of species boundaries is in a group of economically important trees that are very likely hybridizing. And these are species, members of the genus Diospyros, endemic to the Mascarene Islands. So the Mascarene Islands are a group of volcanic islands off the coast of Madagascar. There are 13 extant species endemic. Rodrigues is the oldest island, harboring a single species, while Reunion is the youngest island, also harboring just a single species. The vast majority of the diversity in this group, 11 extant species, there's one species that was thought to be extinct, but 11 of these species all co-occur on Mauritius. So it's harboring the lion's share of diversity. And these species can be morphologically differentiated on the basis of their fruit calyx. So that's the structure sitting on top of the fruit here. And as we'll see later, leaf characteristics. So in many cases, though, it can be difficult to tell these species apart in the wild. And as we'll see, we'll take a look at how the genetics work on that. In addition, many of all but of the 13 species, I should say, 12 of them are a clade, so each other's closest relative. And this is because there is actually 2 colonization events of Diospyros from Madagascar to the Mascarenes. The first one likely started from Madagascar to Rodrigues, which then subsequently likely dispersed from Mauritius into Reunion. And this resulted in the Mascarene Forbesy group. So this is 12 of the 13 species are here. There was a second colonization event from Madagascar, this time just directly to Mauritius, that resulted in just one other species, Diospyros tessellaria. But for the purposes of this talk, we're not really going to mention it too much because it's pretty distantly related to the remaining ones. So for those of you who may not be very familiar with the genus Diospyros, I wanted to quickly kind of give you an introduction, tell you about how how diverse it is and how you might see it in your daily lives. So the genus is incredibly diverse with an estimated 860 species with pantropical distribution. Most of the diversity is found in Madagascar. This picture was actually taken by my collaborator, George Schatz, in Madagascar. All of these species were collected in just a single day trip of hiking around. And it really highlights the diversity we see in their calyx morphology. However, you may be more familiar with the genus in the form of its edible fruit, the persimmon that you can find at your local grocery stores most of the time. However, in the Mascarenes and Madagascar and Africa, it's probably better known for the production of its precious hardwood, ebony wood. This is a jet black wood that's traditionally been used in furniture and musical instruments such as the tuning pegs of violins or the fretboards of violins and the black keys of pianos. And this actually ties into the mascarene species. A number of the mascarene species have traditionally been harvested for their ebony wood, so are of conservation concern. As I mentioned, one is thought to be extinct, and 3 are listed as critically endangered, sometimes having less than 100 known individuals. So it's really important for us to be able to accurately characterize species boundaries in this group to know that kind of limited conservation resources are going towards legitimate species. So that's where the questions for this project kind of come in. The first question we tried to ask or answer were, do patterns of genomic differentiation correspond with morphological species limits in the Mascarene Diospyros? Second, do Mascarene Diospyros species experience hybridization? And lastly, how does the evolutionary history of these species relate to the timing of reproductive isolation? So to answer these questions, I went on 2 different collection trips to the Mauritius and Rodrigues, and then overall I collected, or I had samples collected from all 3 islands and all 12 extant species within the Mascarene Forbesii group. So in this case, we would sample fresh leaf tissue and store it in silica gel to be quickly dried and preserve their DNA for later extraction. And of course, I had a lot of help in the sampling of these species. This is Jean-Claude. A lot of the sampling couldn't be done without the help of Jean-Claude, the Mauritian Wildlife Foundation, National Parks and Conservation Service, the Forestry Department, and staff from the Mauritius Herbarium were also a very big help. So when collecting these, we would identify species in the field on the basis of their leaf and calyx morphologies if they were in fruit. And of course, we took our silica dried leaf samples and took them back to the lab. Once these were back at the lab and following DNA extraction, we used a type 2B we use a 2B RAD-seq approach to genotype our samples. So RAD-seq is restriction site associated DNA sequencing. And what this allows us to do is get a genome-wide view of genetic variation without having to sequence the entire genome. So 2B RAD-seq differs from other flavors of RAD-seq in the use of its type 2B restriction enzyme. So this differs in the sense that most restriction enzymes will have their recognition site and make a single cut. In the type 2B restriction enzyme, it will make 2 cuts on either side of the recognition site, excising a 36 base pair fragment. Always a 36 base pair fragment. There's no variation in this length. From there, we will ligate adapters and then amplify the fragments, adding in a sample-specific barcode that allows us to multiplex up to 96 samples. From there, we'll send our libraries to be sequenced on Illumina HiSeq sequencing. And very briefly, the benefits of using 2B RADseq is that one, it's relatively cheap way to genotype, and we don't need to use expensive fragment machines that allow us to select very specific size fragments. The 36 base pair fragment size prevents or doesn't require us to use any of these kind of expensive machines. So once we get back our reads from the Illumina sequencer, we aligned them de novo and then called single nucleotide polymorphisms that were then used in phylogenomic and population genomic analyses. So these analyses then were used to try to look at patterns of genomic differentiation among our samples. And this is a output of a program structure. And what it tries to do is assign individuals to genetic clusters in a way that maximizes Hardy-Weinberg equilibrium. So in this case, I have 3 morphologically described species, and each genetic cluster is represented by its own unique color. And on the x-axis, we see here, each bar graph is an individual. And the mixture between the 2 colors are representative of the inferred ancestry of in this case, the 2 genetic clusters. So you may notice there's only 3 species here, not 12. And the reason is simply that displaying 12 colors gets a little overwhelming to the eyes. And we see very similar patterns across all of our species. So just showing 3, I think, is fine. We see here largely that these genetic clusters correspond with our morphological species. However, we do see individuals that appear admixed and that likely represent hybrids. We can also look at this genetic variation in a PCA. So this is colored the same way. So up in the upper right-hand corner here, we have Egertarm colored red, Melanita in this dark orange, and Leucomellus in this yellow. And I colored the hybrids here, or the individuals that were identified as hybrid in our structure, and they appear intermediate between the parental genomic clusters. So we also saw morphological evidence of hybridization in this group. And just as an example, the best, clearest one is between Melanita and Leucomellus. So looking at the individuals that appeared admixed in our genetic analyses, their leaves appeared intermediate between the 2 parental species. And this can be seen in Melanita, which is characterized by having a very long, slender petiole, and there's no coloration to the venation here. And then we can compare this to Diospyros leucomellus, that has a very short, stout petiole and a kind of reddish, purplish venation. And then compare this to our inferred hybrid individuals that have kind of an intermediate length petiole. It lacks the coloration of Leucomellus. Venation and coloration is more similar to Melanita, but the leaf base is a little more cordate, similar to Leucomellus, hence very intermediate. To summarize the kind of patterns of hybridization and gene flow across all the species that we saw hybrids between about 7 different pairs of species. And in most cases, we saw relatively few hybrids. So in here, we usually saw maybe about between 2 and 3 or 4 individuals that appeared admixed in our structure, and as well as our PCA, and we also did a hybrid index. The exception, of course, is Egertarm and Leucomellus that had a large number of hybrids. So relating this back to our stages of speciation, for most of the species, we actually saw relatively few hybrids between any 2 species. However, we did see hybrids between many different pairs of species. So I would say, they might fall out around here. So, they're pretty well diverged from a genomic standpoint. However, they do have pretty occasional hybrids with the exception, of course, in Egertarn and Leucomalus. So, however, that's not really the whole picture. Kind of by looking at patterns of hybridization as they relate to the evolutionary relationships of these species, we can begin to address the question of how long it takes to achieve reproductive isolation in this group. So this is a Bayesian phylogeny. It's time calibrated with, in the x-axis, we have time in millions of years. And these are our 12 species. They fall out pretty much into 3 different clades. It's blue, purple, and red clade. And if we map our patterns of hybridization on, a pattern begins to appear. So if you recall, we have Leucomalus and Melanita. We saw evidence of hybridization there. If we map this onto the rest of them, we see that hybridization appears to be mostly limited within these clades. So very little to no hybridization across clades. So by looking at the age of these clades, we see that they're between 3 and 4 million years old. And so we can say that's about how long it takes for reproductive isolation to occur in this group. This is actually similar to some other results we saw in more temperate woody species. So there are some remaining questions I have that just someday would like to answer. One, I'd like to know, what are the ecological drivers of divergence in these species? So I do know that these species, a lot of them occur on kind of somewhat different elevational bands. However, a lot of them still co-occur at different elevations. So perhaps partially being separated along elevational gradients, but that's definitely not the whole story. Secondly, what specifically are the barriers to reproduction among these species? So, you know, our admixture analysis suggested, of course, we don't really see hybrids in the wild between species of, that belong to these different clades. However, just because we don't see them doesn't mean they can't actually form. So I'd like to know if there are pre-zygotic barriers there or post-zygotic barriers. And to do that, I think I'd probably have to do hand crosses to test that. That would be cool to do. And lastly, how might hybridization and integration have fueled diversification in this group? And I just briefly wanted to quickly show you a little bit of work that might be hinting how hybridization could be fueling this diversification. So this here is a figure in which I looked at levels of observed heterozygosity, so a measure of genetic diversity in all of the species that we were just looking at, our 12 Mascarene Forbesii group species. And we see that species that are co-occurring on the same island, so these are all of our Mauritian species, display significantly higher levels of heterozygosity than our 2 solitary island endemics. If you recall, Diospyros diversifolia on Rodrigues is the only one there, and Diospyros bobonica on Reunion is the only species of Diospyros on that island. So kind of what I'm thinking is that in these cases, the Mauritian species have many opportunities to hybridize, of course, within their clades, and perhaps likely as we've seen in their evolutionary history, which could be, you know, increasing genetic diversity that could then be drawn upon for maybe adaptive diversification. So now shifting gears, I just finished talking about kind of a group of closely related tree species, but now I want to be talking about kind of patterns on a much larger scale. So in the tropical Andes, I was looking at patterns of diversity at the level of entire forest communities. So the Andes, of course, are a very important biodiversity hotspot, perhaps the hottest biodiversity hotspot. They contain 15% of Earth's plant species in an area that's about 1% of Earth's land mass. And a lot of this diversity is due to the variety of habitats that we see along its slopes. So this is an image or depiction by Alexander von Humboldt, who was the first to kind of characterize and write about this. We see that forests change along the elevational gradient. For to put this in kind of a cartoon form, where each shape here might represent a species, the composition of these species at different elevations change. And we can visualize this another way in terms of turnover across elevations. So what we're really seeing is a high degree of species turnover across these elevations. And we can visualize this turnover kind of using this graph here. So on the x-axis, we have species turnover. This is measured by Sorensen dissimilarity index. Or on the y-axis, we have that. And on the x-axis, we have difference in elevation. I want to emphasize here, this is not strictly elevation. This is the difference in elevation between communities. So these dots here represent pairwise comparisons of communities. So in this case here, so communities at 0, they're very close to each other in elevation. We see, however, that very quickly, as communities begin to differ in elevation, we see a rapid turnover in species, such that at about 1,500 meters in elevation, communities are sharing virtually no species between one another. And we can just, see this with our own eyes in these. I'm kind of depicting it here in these two pictures. So this is a low elevation forest, kind of near the Amazon basin. We see it's kind of dominated by palms. And compare this to this high elevation cloud forest. Looks completely different, has a lot of epiphytes. So the questions we asked were, what are the factors kind of shaping these communities? And specifically, how did the uplift of the Andes shape these forest communities? So we know that uplift began around 60 to 65 million years ago, with most of the uplift occurring within the last 30 million years. And this uplift, of course, created new, novel environments to which species could occupy. And by examining the processes by which species fill these new environments can tell us a lot about the forces driving patterns of diversity that we see today. So we tested two kind of non-mutually exclusive scenarios of how the uplift of the Andes led to the assembly of these forest communities. So in the first scenario, we have a situation of a recent adaptive diversification, where these new environments are being created and species are adapting to them and diversifying. So we have a scenario in which maybe you could have lowland species occupying and diversifying within these new habitats. Now, if we look at the evolutionary relationships of these species, we might expect to see a pattern like this, where communities are made up of species representing multiple clades that span the elevational gradient. So I should note that here, this dashed line denotes the beginning of uplift. So these are pre-Andean clades. And this is important because we want to look at clades with respect to the origin of the elevational gradient, aka uplift. So we can also translate this into expectations of turnover. So on the x-axis, we have, or on the y-axis, we have a proportion of turnover. And in this case, we can decompose total turnover and look at it in terms of within clades versus among clades. And on the x-axis, we have similarly difference in elevation, just as we saw previously. So under this scenario, we would expect that, no matter whether communities are very close to one another in elevation or very far away from one another or differ a lot in their elevation, turnover between these communities should be dominated by turnover within clades. And we can kind of visualize this. So, in this case, in the green clade, we see there might be different species, but this turnover is all within the green clade, the yellow clade, and the blue clade. Under scenario 2, instead of species adapting to these new environments created by uplift, we can see species that are pre-adapted to these environments immigrating to fill them. So in this scenario, this is one of kind of niche conservatism and ecological sorting. So an example of this, maybe not on the forest community level, but at a clade level, is in Andean lupines. These are, in the Andes, the genus Lupinus is restricted to high alpine elevations. And this work by Hughes and Eastwood found that they originated from a dispersal event from temperate North America. So it's easy to see how species from maybe temperate conditions might be pre-adapted to cooler, high elevation alpine environments that are found in the Andes. So if we look at the evolutionary relationships of species within communities, we might expect a pattern that kind of looks like this, where communities at each elevation are made up of species representing just a few pre-adapted clades. And we can translate these expectations to our expectations of turnover. And they would look entirely different from our expectations of scenario 1. So in this case, for communities that are very close to one another in terms of elevational distance, they're not very different, we would expect most of the turnover to be within clades. So in this case, if we envision ourselves in this community here, in the green one, as we're moving along up the mountain, say, we're going to be seeing mostly things within this clade, would be the expectation. However, we'll get to a certain point that we'll be crossing into a niche or that's another pre-adapted clade might occupy. And in that case, we see an inflection point, in which case among clade turnover would come to dominate the difference in elevation. Okay, so in order to test these two scenarios, we utilized not one, but 2 forest network plots that span the entire elevational gradient of the Andes. So we use plots in Madidi National Park, managed by the Missouri Botanical Garden in Bolivia, as well as ABERG plots located in Manu National Park in Peru. Together, these are 567 plots. And as I said, they go all the way from the Amazon to the tree line. So they cover the entire elevational gradient. And there's actually 2 different size plots. There's 1 hectare and 0.1 hectare plots, which we refer to as large and small plots. In the large plots, people, not me, very great field workers measured and identified all woody plant species in our large plots equal to or greater than 10 centimeters diameter breast height. And in our small plots, they measured DBH equal to or greater than 2.5 centimeters. So all these woody plants were identified to species or morphospecies. And in total, we found around 1,300, these are taxonomically valid species in our large plots, and 1,700 taxonomically valid species in our small plots. So once we got our list of species that occur in these plots, we then wanted to look at the evolutionary relationships and define our clades that we work with. So we mapped the species that occur in these plots onto a Smith and Brown's seed plant phylogeny. This is a mega phylogeny that's time calibrated. So we mapped them all onto this phylogeny and then denoted 2 separate times in essentially cutting the clades at 30 and 60 million years old. So this is kind of cutting where the stems of the clades intersect at 30 million years old. And these denote, in 30 million years old, these correspond to the times in which most of the uplift of the Andes occurred. In the 60 million year old clades kind of represent a more conservative age for that, where uplift was just beginning. So we had essentially 2 different data sets, 30 and 60 million years old, that we then used to look at turnover in these plots and how much of that turnover was either within or among these clades. In addition, of course, we wanted to make sure that the observations weren't due to chance. So we created a Null model that involved tip randomization. So in this, we removed the effect of phylogeny to see if patterns can be explained by chance alone. So we randomly assigned species to the specific phylogeny, doing this about 1,000 times, and then used these data sets and compared them to our empirical analyses. So these are our results. This is for large plots. However, we found very similar results regardless of whether we used 30 or 60 million year old clades, and regardless of whether we used small or large plots, or whether we used morphospecies or didn't include morphospecies. The results were pretty robust regardless to how we varied those. So what we see here is a pattern that really closely matches scenario to the immigration of pre-adapted clades. So when communities, or in these cases plots, are very close together and don't differ very much in elevation, most of the turnover is within clades, as we would expect. But then we get to a point in which we're moving into maybe an elevational band occupied by another pre-adapted clades, and most of this turnover becomes among clades. Compare this to our Null model, and we see that it's quite different. So these results are more than just expected by chance. And overall, we kind of see that this result kind of relates to the idea that it's easier to move than it is to evolve. So this actually has some potential consequences or impacts for conservation. So in this, by finding support for scenario 2, we're inferring that these are, there's a lot of niche conservatism going on, and species are likely to track their environments in the face of environmental changes. So for example, in climate change, we might expect the elevational bands to which they might be pre-adapted to shifting upward. And species at the very highest elevations might not be able to track these changes, and thus might be at a very high risk of extinction. So these high elevation communities are ones in which we might want to keep an eye on. So with that, I'd like to thank a bunch of people, Jean-Claude Sabathian, who helped me in the fieldwork in Mauritius, along with the Mauritius Herbarium staff, Mauritian Wildlife Foundation, Parks and Conservation Service, Forestry Department, all the fieldwork and scientists doing work in the Madidi project, as well as the National Herbarium of Bolivia. And of course, funders, the American Society of Plant Taxonomists, Franklinia Foundation, National Geographic Society, the Garden Club of America, National Science Foundation, and St. Louis University. With that, I'll take any questions. Thank you.
Fascinating seminar, Alex. Thank you. Especially because it gives us those insights in the limitations of the species and also in this plant community assembles. Really interesting. We have questions here in the chat. We have one from Suzanne Renner. She asked, do you think there are more hybrids in trees than herbs?
I would say yes. Mostly because trees tend to have outcrossing mechanisms that I think would promote hybridization. They're also, long-lived. But it's, from an empirical perspective, maybe it's just that more studies are being done on trees. But I would say more likely we would expect to see more hybridization in woody tree species compared to herbaceous.
Hey, Alex. Thank you. I was wondering if you could go back to the slide where you show your results from your final question. Yeah, there we go. That one. Go forward one again. There you go. I was wondering, I don't know, maybe this is just noise in the data, but I was curious, just something that stuck out to me was on the left side in your empirical data, there were this little band that kind of separated off. I guess they're just a mirror of each other, but I was wondering if you have any thoughts on what that might be.
Are you talking about between plots that are very different in elevation?
Yeah, there's yes. So between about 1,500 meters and the maximum difference, there's this data. Yeah.
There's a couple things that could be affecting that. We saw, once we started getting at the higher elevations, there did seem to be more noise, and there's a couple reasons why this could be. One is at higher elevations, diversity is generally decreases. So we're generally seeing less species, which could affect the way the turnover is done. In addition, we also have less plots there. So that could explain the little bend at the very end there. But in terms of those two little bands, I'm not exactly sure, for ones that maybe occur between 1,500 and 2,500. It's funny that you point those out because I actually mostly been focusing at the ones above 2,500, and I didn't actually think about these bands themselves. So I don't have a great explanation for what those could be, or if they're potentially just noise in the data.
Thanks. You never know.
Yeah.
Right. We have another question from Brock Mashburn. He asks, is there applicability of your work in the Andes to understanding the diversification of Diospyros in the Mascarenes? How do Ebenaceae partition the elevational gradient there on such a small scale?
So yes, there is a lot, and I'm currently working on a project in members of the genus Weinmannia that is looking at hybridization across elevational gradients. And in this case, we would typically expect say species that are distributed at different elevations. Say maybe you have a high elevation, I really hope the camera's catching this, I can't see it, and a low elevation, and you might expect to have hybrids at intermediate elevations between there. In this case, the selective forces of the two different elevations may be keeping the morphology of the species largely distinguished. They may or may not have strong genomic differentiation. However, I'm kind of interested in terms of, from a hybridization perspective across elevational gradients, I'm interested in looking at potential transgressive segregation, in which case we could maybe see a scenario where hybrids are able to occupy species outside of either parental species range because they're displaying maybe adaptive morphological features that allow them to occupy maybe higher or lower elevations than their parental species. Hopefully that answers the question.
Thank you. Let's see. So if it doesn't answer the question, then Brock can put another question in the chat. So we have a question here from Michael Moore, who asks, in your Andean forest data set, do you see more within clade turnover when you look at just the less dispersive clades?
Oh, that's a good question. So I guess it depends on what you mean by less dispersive. Either way, the answer is we haven't broken up the data set into species or clades that are very in known dispersal ability. But that would be something interesting to do, and what we might expect to see in that, looking at clades that differ in their dispersal ability.
Great. Here we have a comment from Eduardo Aguirre. He said, super interesting work. And he asked, how did molecular clocks were calibrated for building the phylogenes? Which criteria can be used across all of those clades?
So I assume you're talking about the Andean work we did. So in the mega phylogeny of Smith and Brown, they use a ton of different fossil calibration points to calibrate this large phylogeny. And in our case, we're attaching species to where we know they occur in that phylogeny. However, obviously a lot of them are not going to be in that phylogeny. In that case, they have to be placed in sort of a polytomy related to their taxonomic knowledge. So to answer your question, this isn't like a fully perfectly bifurcating tree with dates between every node or divergence. So in this case, it's more of a rough idea of times of divergence, because in order to build a fully bifurcating tree for these, we'd have to sequence and genotype all of them, and even then, getting genetic data for all of them, we're likely not to get a fully resolved tree. Because of course, as we saw in the Diospyros, in the case of many closer related species that maybe diverged relatively recently, maybe within the last 10 million years or so, you might not be able to tell them apart very well. So the answer is, I guess, the short answer is that we're not using this tree in the sense of in the expectation that it's a perfectly time calibrated tree, but it does give us a very good approximation of the divergence times by using this Smith and Brown's megaphylogeny.
All right, we have a picture. We have a question from Robbie Hart, who asks, Alex, to follow up on your answer to Brock's question, would it be possible to use these methods to track expanding or shrinking hybrid zones across time as climate shifts species elevational distributions?
Yes, I think it absolutely would. And I think that actually be a really cool thing to do. So, we could do an experiment where we look at, different age classes and compare them to different, hybrid classes, because we can determine, give a rough idea of the hybrid class, whether it's like an F1 or backcross individual. So, we could come back over time and sample seedlings and say, well, do we see the movement of certain genotypes from one parent or another parent moving in a certain direction and elevation or something like that? Yeah, I think it's totally doable. And yeah, we could talk definitely more about that. That'd be awesome.
Thanks. Here we have another question from Peter Weiss Jackson. It's an interesting question. He says, with such threatened species as the Mauritius Diospyros, how may one design a conservation program that takes into account adequately the hybridization you have identified?
Yes, that is a very good question. So traditionally, of course, hybrids tend to just kind of be ignored or pushed under the rug, try not to think about them. But I think hybrids represent, a real evolutionary process. I think, I think it would be important to conserve hybrid populations as well, because as we saw, this hybrid populations can actually be, a source of genetic diversity that could potentially help, down the line in their evolutionary history, and potentially combat things like changing climates or environmental changes. That, I should say, there are still things to be taken into consideration. For example, you could have situations where there's genetic swamping, where there's a rare species that is hybridizing with a very common species and is essentially getting backcrossed into oblivion. We would definitely want to avoid situations like that. But with genomic testing and stuff, we can identify these and kind of move forward. So short answer is, I would say hybrids are real and they're a real process that's going on, and you should take them into consideration when doing conservation planning. Specifically, I think protecting hybrid zones and monitoring them in terms of looking to whether these hybrid zones are static or relatively permanent, because there are many outcomes to hybridization, and yeah, that could potentially be important for conservation.
So a little bit on the conservation angle. Well, just thinking about the it's easier to move than to evolve conclusion. I'm curious, is this a pattern that's seen widely or generally across many different species, or even is it sort of different between biomes? You have this perfect example of the Andes, but I'm curious if.
Yeah, I've read some papers that have looked at this as well, looking at kind of niche conservatism in other mountain areas, and they found some similar results. There's one thing I wanted to make kind of clear that I didn't get to make the point of during the talk, is we kind of set this up as the two scenarios are rapid adaptive diversification or immigration of pre-adapted clades. However, it's important to realize that you can still have adaptive diversification within these pre-adapted clades, but they're just with respect to elevation. So for example, let's go back to lupinus. So we see here that there was still a radiation, even though this is all within high elevation environments or alpine environments. So yeah, so I would say, to answer your question, yes, we have seen this pattern in other mountains. Yeah, but yeah.
So following up one of those questions, what are your thoughts on calling species to hybrids with viable populations? So in other words, can we call a hybrid and species if they have these viable populations?
Say that again?
So what are your thoughts on calling species to hybrids with viable populations? So you have these big populations of those hybrids. Can we call species to them?
So you're saying, do you think we should call those hybrid populations species?
Yeah, what are your thoughts about that, calling them species or not, or just calling hybrids?
I would probably not want to call them species, mostly because there are many different possible outcomes to hybridization, and you can have homoploid hybrid speciation occur. That could be one outcome of these hybrid populations, but that generally seems to be pretty rare. And in many cases, we might see the hybrid zone disappear over time if we have strong divergent selection on the parent populations, and we might see the buildup of co-adapted gene complexes over time, to get to the point where, you know, hybrids might be selected against, because, you know, their hybrids would be in the hybrid individuals might have recombination that destroys these co-adapted gene complexes. So the point is that we just don't know oftentimes what the outcomes of hybridization are, and we see that there's many different outcomes. So I think to call it a species might be premature, because there's so many things that can potentially happen.
All right, we have a question here from Sebastian Tello, who asks, would it be easier to evolve than to move for species that hybridize? Is there evidence that the genetic diversity resulting from hybridization facilitates adaptation?
Yes, and well, that's the thing. There's a lot of studies I see that talk about this. How hybridization, you know, creates genetic variation that can be used to you know, spur a genetic or a adaptive radiation. However, there's actually relatively few cases that actually are like proving this. So take, for example, in Diospyros, we see that there is hybridization in the ancient past, using a d-statistic analysis I did. I didn't really mention it because I didn't want to go too deep into it, but we see that, you know, there's been hybridization in the past. So a lot of people might infer that hybridization may have been very helpful for the diversification of a particular group. But to actually test it, I haven't seen anything actually testing whether, you know, novel variation could be used on to you know, spur an adaptive radiation. There is evidence of, you know, transgressive segregation, in which case that is maybe as close as you could get. Lauren Riesberg, of course, in his sunflower work, looking at salt tolerance, where, you know, hybrids were more able to experience salt tolerance than either parental species, and in that case, you could see an argument for that. But there's been pretty little experimental evidence so far to show that. It's mostly been observations of existing patterns of diversity and making inferences.
Okay, here's a question from Yusan Yang. She asks, a bit parallel to Sasha's question, is the quotation, is easier to move than to evolve apparent for moving diversification across latitude as well? For example, temperature versus tropical?
Oh, that's a good question. Latitudinal diversity gradients. I would expect similar things, and I'm sure work has been done on it, but I'm not that familiar with that work. So I don't know. Sorry.
Okay, no problem. We have some more questions, but unfortunately it's time to go. It's 5 o'clock. Alex, thank you for your time and for sharing your fantastic work, really interesting results. And thank you everybody for attending this week's seminar, and stay tuned for the next Live on Earth Collaborative seminar series at the same time. Have a good day.
Thank you.